I'd like to welcome everyone to Health Coach Seven's Methylation and Hormones. And I wanted to thank you for your attendance. We had some great attendance here on our webinars. You probably remember starting off with the putting bad genes on good behavior. And by the way, just wanted to mention to you that I'm in the current movement of writing two books. One is Putting Bad Genes on Good Behavior, and the other one is Protecting My DNA, Reprogramming for Better Health. And those are two books that'll be, that are in the works that'll be coming out soon. You probably spent some time looking at, the schedule, and I just wanted to demonstrate to you. You probably see my screen sharing here. But I wanted to go on to the schedule really quick to show you, to kind of just go into what we have been covering. And we had some very interesting segments, starting off with putting bad genes on good behavior. And then you can go to the Health Coach 7 Facebook site, or you can go to Health Coach 7 website, www.healthcoach7.com. And as you can see here, I pulled it up here on Facebook, and it's we did Putting Bad Genes on Good Behavior. The session 1 was Genes and Methylation. Second session was Metabolism and Methylation. Third was Immunity and Methylation. Fourth was Immunity, Inflammation and Methylation. Fifth was Detoxification and Methylation. We're going through those dynamics. And Dr. Jeff did session number 5 and did a fantastic job outlining the detoxification. You can go through each one of these segments. We'll be reposting them for your enjoyment, and you can future listening. They're packed with a lot of information and a lot of articles there that we shared. We actually make available, and so you can actually have the opportunity to peruse those articles as you have the time to do so. So this segment is about hormones and methylation. Now, hormones, a lot of the times we often, forget that the importance of hormone regulation is an overall mechanism of metabolism. And so what we want to be sure that we don't ever forget is that when we look at steroidal hormone principal pathways, as you can see here, the interesting thing about this is, I'm enlarging it a little bit so you can see it, is that it's very complex. So as we look at the methylation cycle, methylation and integral steroidal hormonal pathway regulation are very much interconnected. And as you can see here, everything starts off with cholesterol, and you got the intake input from dietary fats, and you've got cofactors that come in here. Vitamin B5 is one of those. And then we've got the cholesterol as the moiety or the matrix that actually starts funneling into the production of every hormone that is so essential for our body's need. And you can see cortisol here. Some of you might be very familiar with cortisol, as it's very much in regulation of stress and stress patterns, and keeping body immunity stable. And a lot of the times you probably heard of adrenal insufficiency or adrenal stress. We often go to this part of the pathway that actually is regulated. DHEA falls into that category as well, which is right there, and the DHEA sulfate, which is a cholesterol sulfate moiety. And you can actually look at this and you can see how other of the hormones of the estrogen family and testosterone family start coming into existence. And you might recognize some of these, including the estrogens as well as the progesterone, which is actually on this side here. Notice that the progesterone feeds into the cortisol pathway, so it can actually be very beneficial in adrenal fatigue. So that's why when it's given as a replacement therapy, especially for some women with a lot of menstrual difficulties or cycle irregularities, they also notice that their energy actually gets better. And we had a very interesting question about a person listening to the seminar. My progesterone levels are elevated, and when I take it, it causes my cortisol levels to go up higher. So actually, the progesterone she was taking, I'm sorry, progesterone, she was taking progesterone. There was prescribed progesterone, and I wasn't certain if whether or not her progesterone levels were already existing as being elevated, but she was taking progesterone as prescribed with the bioidentical hormones, and then when she discovered that it was feeding her cortisol pathway, which of course it does, but every time she took the progesterone, she would have these aggravating symptoms, which probably was definitely over revving her adrenal system at that point, and then also creating some methylation overcharge, maybe, and maybe definitely creating that crisis type of symptoms that are associated with maybe over energizing the methylating pathways. So anyway, you can see the relationships here, very, very important. And, as we look at hormones, we want to be sure that we, see that methylation in hormones, there's approximately 1,441 genes listed on genecards.org that actually are regulatory genes that are responsible for hormonal control. And you might recognize some of them here. I mean, you've got estrogen receptor 1, which you probably recognize. You've got sex hormone binding globulin, which you recognize, thyrotropin releasing hormone. And as you scroll into and you do the search, progesterone receptors there, you're going to see a lot of the associated hormone gene controls very integrally involved with the methylation cycle as well. And then you got methylation in neurohormones, and there's approximately 61 gene controls that are firmly involved with methylation and neurohormonal regulation. And you can see serotonin is there, probably the HTR1A and 2A, PTGS1, which is controlling inflammatory responses, and you've got many, many others as you can go through here. And you can see on the next page, it'll give you another outline of some of those neurohormonal and methylation relationships. And then we have methylation and neurotransmitter relationships, which we can see is a lot more involved with 361 hormones as part of the regulatory movement there in cross relations. COMT you can see is there, which is the catechol-methyltransferase, which a lot of people have concern about, positive COMT and positive DRD2. You're going to see MAOA, MAOB. A lot of these here you're going to peruse, you probably see a lot of neurotransmitters that you recognize. Some of the hormones that definitely affect methylation cycle, and stress in itself can affect the release of these hormones or the impairment of the efficiency of operation of these hormones. As you can see, they're listed here: gastrin, secretin, cholecystokinin, and gastric inhibitory peptide. All of these are very important hormones in regulating gastric movement, digestive response, and setting up the whole scenario for proper digestion. And a lot of the times we really don't recognize how these GI hormones can actually facilitate more stress reactions in the body and actually facilitate maybe methylation disturbances because of distress. And so you got to be looking at GI hormones as well as the other hormones that we just mentioned. The important thing is to remember is that when we're looking at methylation and we're looking at control of hormonal metabolism, we got to remember that the metabolism as a whole, this is the human metabolic map, and methylation is actually a portion of an integral part of metabolism. This is the human metabolism map. And if you were to look at where does methylation exactly fit in the process of metabolism, so you can see the generation of DNAs and RNAs and the actual activity of a lot of neurotransmitter and catecholamines, which are all here, a lot of methyl activity going on and feeding into the interdependent breakdown of glycolysis. So if you were to really place the methylation map in here, you would probably have to fit it in, I would say, in this segment right in here. That's a kind of a portion of the methylation because you got the Krebs cycle present, and you've got down here, you've got vitamin D metabolism. Hormone metabolism is right over here, as you can see in this lower hand corner. and then you might recognize some very important segments in methylation, which is the methionine, homocysteine, cystathiamine, and the glutathione. So if you're looking at it from the standpoint of where it's all going to fit, you've got methylation, integral part of metabolism, explaining from B, C, down from B, you can start from B on the map all the way down to H and across to, right about, wow, it's a significant part. You might as well just say that all of it's involved. And so anyway, methylation is an integral part of metabolism, and we need to remember that it fits into this map. So, and it controls and it has influence in several parts of this metabolism. So if we were to put it into a major place, I think we would probably be fitting it into this DNA RNA pathway, which will then, also be fed and influenced by the movement to coming off of the Krebs cycle and into the neurotransmitter relationships and things like that. So anyway, over here on this side, we're actually seeing the relationship of the steroidal hormonal pathways that I just mentioned. And you can see it's very important for us to know that when we're looking at hormonal interactions, we want to be looking at checking salivary levels, maybe salivary blood and blood levels, maybe, and also urinary levels. I would say salivary, urinary, and blood would be a good way to assess where your hormones are, and usually deficiencies and excesses will definitely counteract the efficiency of methylation and the protection of ultimately going on the other side, regeneration of RNA, DNA, and RNA. So it's very important for you to realize that if you want to be cycling some good movement in this area here, the methylation cycle has to be energized and moving appropriately down here, and it has to be moving appropriately in the interface in the Krebs cycle and the control of glycolysis. So it's just really important to know that we can't take methylation out of the metabolic pathways. We got to make sure that we're discussing them as a integral part of moving to facilitate better DNA regeneration and protective cell health. So it's very important there. And then, of course, the growing role of methylation in hormonal control of DNA. Very good article here that actually discusses the endocrine relationships of glucocorticoid responses and general effect of many nuclear receptors on hormones. And you're going to see how the CpG dinucleotides are actually affected by the actual movement of hormonal balance. So I think you're going to find that a lot of this is a lot of verbiage, of course, but it talks about the existence of hormone-dependent, interdependent tumors being a substantial problem for the treatment of several endocrine tumors or hormone antagonist issues, and hypermethylation can selectively silence those multiple promoters of steroid receptors. So you can actually see that hypermethylation has got indications in several types of cancers, and you can actually see aberrant DNA methylation promoter regions in estrogen, progesterone, androgen receptors of course, the testosterone origin. And it's very, very important to see that hypermethylating patterns are often seen in tumor promotion that are hormonally fed, if you will, because they're receptor-positive hormone tumors. But it's really, when we're talking about just general function of hormones in general, we want to be careful that we look at any disturbing factors that might promote faulty metabolism or faulty detoxification through the liver and through the kidneys. You got to remember that the detoxification pathways, if they're congested and they are not working appropriately and changing toxic metabolites into non-harmful metabolites to be able to be excreted, it's very essential that that pathway be energized appropriately and that it not be bottlenecked, because what winds up happening is you have the lingering effect of toxic reactants that can come from hormones that are not being appropriately metabolized. And there are a lot of cofactors involved in that. For instance, in estrogens, you've got calcium D-glucarate. That's a very important bionutrient to actually assist with estrogen metabolism and detoxification and movement. You've got indole 3 carbanols that are essential in being able to help metabolize the estrogens appropriately. You actually have to be sure that your adrenal system is working appropriately to be sure that thyroid and adrenal connection and stepping up the metabolism so that these hormones are actually working efficiently in the appropriate temperature environment and an appropriate climate of stress. So you got to be looking at all these things integrally. One cannot be, you can't neglect one uh, and not expect it. If you overlook it, you would wind up finding that it could be the missing link as to why the hormonal balance is disturbed in general. So methylation and hormonal controls very much integrated together. And the fact is, with the hormones, with hormone relationships, whether it's neurohormones or steroidal hormones or any type of neuroendocrine pattern, which is a neurohormonal interaction, you're going to find that the hormone dynamic is probably one of the most powerful dynamics in the prevention of premature aging. So, like in this, ultimately speaking, the integrity of the cells. When we actually look at integrity of cells, we want to be sure that we're looking at the overall protection of these cells. So, for instance, if we decided that we wanted to do ESR, which is an estrogen receptor, we go to estrogen receptor 1 and we say, okay, well, estrogen receptor 1 is responsible for the sensing uptake of estrogen. As you can see here, it's very much involved in the receptivity of essential sexual development and reproductive function, as it says there, but it also plays a role in other tissues and bone and the process of moving in cellular expression to be sure everything actually affects the cell expression. So when you actually go down to movement into seeing where exactly we'll define the ESR movement, as you can see in the compartments, the Golgi actually has a lot of packaging right there. The nucleus actually is definitely very active in this particular gene control, and you've got it very much distributed in the cytosol. So estrogen receptor positive cancers are pretty much said to have a very aggressive cyclical cycle in development, which means that the cells replicate faster with the expression of estrogen receptor on the surface, because the estrogen then binds to the surface of the cells and it energizes the nucleus for an accelerated cycling, what they call a cell reproduction cycle advancement. So it actually triggers that cycling, and that's the reason why the cells become more, they reproduce themselves quicker, because they're hormonally influenced. And things like DIM help with the residual estrogens that actually would have negative effects on binding. And the calcium D-glucarate would actually facilitate the detoxification of excess estrogens in the tissue. And there's also ways of doing that with progesterone and testosterone. Okay, so you got to be looking at these pathways appropriately. Got to be looking at your hormone balance appropriately. You know, the issues with hormones is this, basically. If you have deficiencies, it's a predisposition to create some kind of a negative end result in disease progression. And cancer can be facilitated by uh, I would say, efficiency of hormone balance. And then the overproduction is the other extreme. And then you can wind up having disease associated like cancer with excess and the actual effect on oncomutations in cells that have actually expressed those hormone receptors. And you have to remember, a clean, healthy cell that has an intact nucleus with good engineers will not have expressive receptors on the surface of the cell that could be detrimental to the cell's well-being. Okay, you got to remember that cells actually grow and reproduce themselves at a very, very fast rate. So bad cells can reproduce themselves. Any kind of oncogenetic cell, oncogene, develop a signal cell can actually reproduce itself very quickly and mutate very quickly. So with that, thank you for your attentiveness, and I hope that this has been a very interesting presentation for you. And I look forward to seeing you on the next presentations that are coming up here, very interesting ones. You can go on Health Coach 7 Facebook site and see them. And these recordings will be reposted, and you can listen to them again and again. Again, I would strongly suggest that, remember, methylation and hormones, there should be the correct evaluation of making sure knowing where your hormones are, and any kind of strategic therapeutic approach should always be developed in a personalized way. One size doesn't fit all in this, and cofactors need to be addressed, and detoxification factors need to be addressed to be assured that the balance of hormonal function is working favorably to sustain your longevity and prevent premature aging. And it really makes a difference. So look forward to seeing you next time. Thank you for your attentiveness, and we'll see each other again. Okay, take care. Bye.